So good morning. I will be talking about this article, comparison of adaptive response of Streptococcus aureus versus Streptococcus mutans and the development of chlorhexidine resistance. So this was published in 2002, 19 May, and in the journal Frontiers Biology with impact factor of 4.6. So firstly, we'll talk about what is chlorhexidine. Chlorhexidine is a broad spectrum, gold standard antibacterial mouth rinse that acts by disrupting the bacterial cell wall, causing intracellular leakage. So mild exposures of chlorhexidine to the bacteria can lead to cell deformation, whereas the severe exposure can result in a bacterial cell death. So increased use of chlorhexidine has caused a rise in antimicrobial resistance bacteria, which with some developing cross resistance to antibiotics. Means the different species are acquiring the antimicrobial resistance to chlorhexidine. So genes for AMR have shown to transfer between bacteria, triggering genetic changes that promote multi-drug resistance in pathogens. So antimicrobial resistance is uncommon for oral bacteria because due to the limited biofilm penetration. So the drug do not penetrate the biofilm, it does not go deep within, so the resistance is hard to occur. But the CHX, that is absorbed by the soft tissue, creates a prolonged exposure and causes the potential to develop resistance. So to prevent resistance, the use of CHS is restricted to essential medical application. So understanding why some oral bacteria remain susceptible, whereas some bacteria develop resistance is crucial. As the title suggests, there were two bacteria that were tested. So the Staphylococcus aureus and Streptococcus mutans. Response to chlorhexidine was understood focusing on the cell wall damage, membrane integrity and metabolic activity. So they used fluorescence microscopy and metabolic assays. The research aims to determine why Streptococcus aureus can develop chlorhexidine resistance while mutants remain susceptible. So the materials and methodology. Firstly, they grow bacterial strains and harvesting. So two green fluorescent bacterial strains were used, Streptococcus mutans UA159 GFP and Staphylococcus aureus ATCC 12600 GFP. So these two strains were used in this study. They were grown in specific media. For Streptococcus mutans, the media was Todd Hibbert broth, and for Staphylococcus aureus, the media used was Krypton soya broth. And so the bacteria were cultured, washed and resuspended in the phosphate buffer media and adjusted to a concentration of 3 into 10⁸ cells per ml for this experiment. So minimal inhibitory concentration for chlorhexidine was found. So the commercial mouth rinse for this commercial mouth rinse was used containing 0.12, that is 1200 microgram per milliliter chlorhexidine. So serial dilutions were done for chlorhexidine and test was done to determine the minimum inhibitory concentration, defined as it is the lowest concentration that prevent visible bacterial growth. So the development of chlorhexidine resistant genes or variants. So the bacteria was cultured with increasing chlorhexidine concentrations over multiple days. So the resistance was assessed by measuring changes in the minimum inhibitory concentration over a period of time, and the resistance development was confirmed in Streptococcus aureus, but it was absent in the Staphylococcus aureus, and it was absent in the Streptococcus mutans. So the surface enhanced fluorescence. This technique was used to measure the bacterial cell wall deformation upon chlorhexidine exposure. So the bacteria were adhered to a metal surface in a flow chamber, and the fluorescent changes were monitored over a period of time to infer the cell wall integrity. The bacterial membrane damage was also assessed. So Propidium iodide staining was used to assess the membrane permeability. Fluorescent microscopy quantified the percentage of bacteria with compromised membranes after chlorhexidine exposure. So after the exposure of chlorhexidine, they quantified how many bacteria have compromised bacterial membrane using the fluorescent microscopy. The metabolic activity of the bacteria were assessed. So in response to chlorhexidine, it was evaluated using the MTT reduction assay. Absorbance at 560 nanometers was used to determine the bacterial metabolic activity before and after the exposure of chlorhexidine. Physiochemical bacterial cell surface characterization. So hydrophobicity was assessed using the microbial adhesion to hydrocarbons MATH assay. The zeta potential measurements determine the bacterial surface charge properties. Lastly, the statistical analysis was done using paired and two-tailed students t-test with using Excel, and the significance was established less than 0.05. So the results of this study was found to be as follows. The chlorhexidine resistance development in S. aureus developed resistance. The S. aureus developed resistance when exposed to increasing concentrations of chlorhexidine over multiple days, with the minimum inhibitory concentration increasing to 4 times, that is from 1.2 microgram to 4.8 microgram. So earlier, before the resistance development, the chlorhexidine that was required to kill was 1.2 microgram per ml, that increased to 4.8 microgram per ml. In S mutants did not develop resistance under the same conditions and could not survive beyond its minimal inhibitory concentration. So these changes was noticed in both the bacteria. The cell wall deformation upon adhesion of and chlorhexidine exposure. So Staphylococcus aureus showed gradual and irreversible cell wall deformation upon exposure to chlorhexidine, indicating continuous structural weakening. Chlorhexidine resistant S. aureus variant exhibited even greater deformation, suggesting that an adaptation mechanism that involves structural modifications. And S. mutants, in contrast, displayed rapid but reversible increase in cell wall deformation upon chlorhexidine exposure, indicating a self-repair mechanism. The cell wall damage and membrane permeability. The S. aureus exhibited significant membrane damage with up to 50% of the cells allowing penetration of Propidium iodide after chlorhexidine exposure. The resistant S. aureus strain had reduced membrane damage, suggesting that the resistance mechanism involves reinforcement of the cell wall. The S mutants initially showed widespread cell wall damage but almost completely self-repaired within 2 hours, preventing sustained membrane permeability. The metabolic activity and efflux mechanism. The S aureus exhibited an increased metabolic activity after chlorhexidine exposure, suggesting activation of energy-dependent resistant mechanism for like efflux pumps. Whereas in S mutants, despite experiencing initial cell wall damage, it did not show any increase in metabolic activity, indicating lack of resistance related efflux mechanism. Physiochemical surface properties. Neither of the species showed change in the zeta potential or hydrophobicity, indicating that chlorhexidine resistance is in S aureus is due to altered surface change, is not due to altered surface change or adhesion properties. So, coming to the conclusion for this, the S aureus responds to chlorhexidine induced damage by activating efflux pumps, which prevent cytoplasmic accumulation of antimicrobial agents. This allows the subpopulation to survive, adapt, and eventually develop resistance. In contrast, S mutants undergo self-repair when exposed to chlorhexidine. While this mechanism protects the cell wall, it prevents efflux of chlorhexidine, leading to cytoplasmic solidification and cell death. So, in this explains why Streptococcus mutans does not develop the resistance, while the S aureus develops the resistance. The strengths for this study was as follows. They did comparative analysis, so directly comparison was done of S aureus and S mutants to explain the differences in chlorhexidine resistance. The use of advanced techniques such as surface enhanced fluorescence for a precise measurement of cell wall deformation. Real world relevance. So these findings can be applied in clinical settings, particularly in oral healthcare. The multifaceted approach. This investigates the bacterial response from multiple angles, including the membrane damage and metabolic activity. Unique mechanism was identified, proposes the suicidal self-repair in S mutants as a resistance preventing mechanism. The limitations for this study: the limited bacterial strains were used. Only 2 strains were used, as it said in the title. So then it was an in vitro study, and it may not fully replicate the oral conditions in vivo. Medical constraints, methodological constraints. The measures bulk bacterial population, potentially missing the single cell variations. Short-term focus. Primarily, it investigated immediate response without the long-term resistance assessment. Lack of genetic analysis. This study does not include the genetic studies to confirm the previously mentioned molecular pathways. Thank you.